genetic code. So we know DNA made up of two polynucleotide strands, and these long chain polynucleotide strands, they are made up of nucleotides. Nucleotides are made up of uh, base, nitrogenous base, then sugar and phosphate. So nitrogenous bases are adenine, guanine, cytosine and thymine. So these nitrogenous base bearing nucleotides, they come together, and on another strand complementary nucleotides, they are placed, and two polynucleotide strands, complementary strands, they come together and forms a DNA. So when DNA is responsible for, we all know, DNA is responsible for the transfer of character. Actually, a particular segment of DNA, which is functional one, is responsible for the transfer of character from one generation to another generation, particular character. So like these segments, many segments, this segment you call it as a gene, and such many segments, functional segments, they function for heredity. So how it happen actually? The DNA, particular functional segment of DNA, that gets separated, and on one of the strand of DNA, mRNA is produced. This is the process of transcription. So due to transcription, production of mRNA takes place, and these mRNAs, they carry the information ki what type of protein should be produced during protein synthesis on ribosomes. So we are going into the details. One should know that protein is made up of amino acids. These amino acids, they come together in specific sequence and in specific number, and that determines the type of protein. So in between amino acids, peptide bonds are formed, and this is nothing but the protein. So different types of proteins are produced as a result of transcription, that is formation of mRNA on the template DNA. Now, who is responsible for gathering the amino acids in a protein in a specific sequence? Actually, that thing we are going to study in the chapter named as a genetic code. So how one can define genetic code? So in brief, we can say is a system of signals for genetic code is can be defined as a relationship between the nucleotide sequences in nucleic acids, maybe DNA or mRNA, and the amino acids in the protein. Just now I have told you proteins are made up of different amino acids. 20 different amino acids are required for synthesis of protein. Once again we will see the definition, the relationship between the nucleotide sequences in nucleic acids and the amino acids in the proteins. So this is in brief definition of genetic code. The base sequences in DNA or in mRNA code, or specify the amino acids of protein. Base sequences in DNA or in mRNA code specify the amino acids of the protein during protein synthesis. So here we will have to see the possibility ki how many actually we should know there are about 150 different types of amino acids found in nature. Out of them only 20 different amino acids are required for the protein synthesis in the cell, and 20 different amino acids take part in the synthesis of the protein in the cell. And there are thousands of proteins in the cell, and majority of them are enzymes. So one can say all enzymes are proteins, but all proteins are not enzymes. Proteins present in a cell differ from one another in the number and specific sequence of amino acids. So that's what I told you. The number of amino acids in a polypeptide chain and sequence of amino acids, these two things, they determine the type of protein. Polypeptide chain contains about 100 to 300 amino acids in a specific sequence of 20 different types of amino acids in it. Now we will have to see how sequence of nucleotides that codes or specify one amino acid, and this sequence, the sequence of nucleotide that codes or specifies one amino acid, we will call it as a codon. So we will have to see the possibility कि probably how many bases they specify one codon. So first possibility, that is a code may be a single letter. If this possibility we look towards, so there are only 4 bases: adenine, guanine, cytosine, and in case of mRNA, uracil instead of thymine. So only 4 amino acids can be specified if we consider this possibility. That is, the codon is singlet, or call it as a singlet codon. So these are not enough to specify 20 different amino acids which must be there in the formation of different types of protein for a organism. So we will discard this possibility. Then the another possibility, that is the code is doublet instead of singlet. Now we will see the possibility, that is a code is 2 letter or 2 alphabets. If we consider this possibility, if sequence of 2 bases specify one amino acid, then you call it as a doublet or duplet codon or doublet codon. If 2 bases code for one amino acid, then only how many possibilities? 4 into 4. That is 16 amino acids can be specified if we consider this possibility, that is codon is a doublet one. So it is not also enough to specify 20 different amino acids. We need 20 specification of 20 different amino acids. So the another possibility, that is the code is doublet one, is also discarded. Now we will have to go towards the next higher rank we can say. Previously we have seen if one take one base at a time, only 4 amino acids can be specified. In the second possibility, the 2 bases at a time, and so only 16 possibilities are there. And now we will think over the possibility what will happen if one consider that the codon is triplet or code of 3 letters or 3 alphabets. So if the code is composed of 3 letters, then call it as a triplet codon. If 3 bases of DNA code for 1 amino acid, such code is called as a triplet code or a triplet codon. If 3 bases of DNA code 1 amino acid, then 4 into 4 into 4, like this, 64 3 bases at a time, 4 into 4 into 4, like this you can say, you can have 64 different combinations which can specify the amino acid. Actually it is more than that of 20 different amino acids. These combinations are 64. But we will have to consider this possibility as the most probable one. And what about the excess codons here? Because only 20 different amino acids are there, and the remaining excess codons, they might be specifying the same amino acid from 20 different amino acids. Same means amongst 20 different, there may be more than one codon for specifying a particular amino acid. So like this, we will assume that this is the most probable answer, that codon is a triplet one. Now here in this chart, three bases are taken at a time. For example, here you see or UCA or UCG and like that. So here take second base, take here first base. Just see once again, this is the first base. On this side you take first base in the form of UCAG. Okay, then take second base here, again UCAG, like that. And the third base here, see third base here, and you take the 4 bases here, UCAG, and in the same manner UCAG here, UCAG here. So first base you take here, second base here, and third base here. So what will be the first possibility? You take first base U here, U, then take second base here, it is U, and third base is also again U. So first combination is U here. So like this, take the combination from this place. So first base C is here, then second base is also C, that is here, and third base is U, so that is here. Like this, prepare all the combinations, and these combinations, which combination or which codon is responsible for specifying which amino acid is shown or written over here. For example, here phenyl leucine you can see here. So the short forms are shown over here. So actually this is a genetic code dictionary given by Indian born scientist Hargovind Khorana. This is a great achievement, and you can say a evolutionary achievement in case of molecular biology, and the prominent and dominant work of Indian scientist who born in India, settled in USA, and he is responsible for giving output in the form of this dictionary. So in detail we are going to study what this dictionary says later on. Now we'll go towards the properties of genetic code or characteristics of this triplet codon. Once again we will see what is genetic code, that is relationship between nucleotide and amino acid sequence in protein. Call it as a genetic code. The hereditary units of genetic code are called as codons. Each codon is formed of 3 nitrogenous bases, which codes for 1 amino acid, and we call it as a triplet code. Code is present in DNA. Codon is present on mRNA, and anticodon is present on tRNA. Some of the properties of genetic code we will see now after taking into account these basic things related with the genetic code. So first property of genetic code is its universality. So code is universal. Also call this property as a universality of the genetic code. Actually, genetic code is established from the studies on bacterium named as a Escherichia coli. But this genetic code is valid for all organisms, as code is found in all organisms. So take example of virus, bacteria, plants, animals. Such property of genetic code is called as a universality, and thus one can say that the code is universal. Genetic code is universal. Second property, that code is triplet, and that thing we have seen in brief how triplet codon is essential. Second property, that is code is commaless. It means on mRNA the nucleotides they are there, and not a single base is wasted to say that now stop or take pause or like that. So continuously it is read. See here, in this case, in this diagrammatic sketch, suppose this is the stretch of mRNA like this. on this mRNA, these are the uh, bases. See, like this, bases. And these bases, for example, here they have shown one of the triplet codon like U, second UCG, like this you can represent on the mRNA. So the same thing is shown here. So bases on mRNA. So these are the bases. So see here the base sequence U, then UCG, UCG, UCA, UCC, ACC, like this. Okay. Now, see, related to the comma-less code, we are saying that genetic code is comma-less. It means no codon is reserved for punctuations, or not a single codon is wasted for saying ki now take pause or like that. In other words, we can say each triplet codon in mRNA is not separated by a comma or a spacer, or not any base is wasted for this thing. The genetic code is always read in an uninterrupted manner from one end of mRNA strand to other end. Just now I have shown how to read from one end to the another end like this, and no single base is wasted in between. So if this is the situation, one should read the sequence of bases on mRNA in a continuous fashion. A comma-less code will not have the comma bases, and it can be represented like this. So here bases on the mRNA, and the sequence of bases we have shown here, and take 3 at a time. For example, first U, and go towards see this U, okay, then go towards the genetic code dictionary. So what codon we have taken? U. See, this is the first one, first combination. See here, U. So here the U codes for phenylalanine, then CUC, where CUC is just CUC, it is here. CUC codes for what? Leucine. Is it clear? First, UUU codes for phenylalanine. Second, CUC codes for leucine. Then search for GUA. G, first base G, second base U. GUA. So GUA, it is here somewhere, just here. And what it codes for? The abbreviation VAL for valine. Valine is the amino acid. So like this, third codon, it codes for valine. Then fourth one, how to search? Fourth one, what is the codon? UCC. First base U, go towards U. Okay. U, second base C. See here, UCC. So where UCC is, just see. So here UCC, and it codes for what? the abbreviation SER for serine. So it codes for serine. And one more is remaining in the base sequence of mRNA, that is ACC. So how to search A? Go for first base A, second base C, A, C. So it will be somewhere here, ACC. It codes for threonine, THR for threonine. So like this, you will have to search in the genetic code dictionary, and accordingly phenylalanine here first is produced in the it is seen in the protein produced. Then second amino acid seen is the leucine, third is the valine, fourth is the serine, and fifth is the threonine. So like this, the amino acids in protein, they are in this sequence, and that is the fact. And thus one can say that the code is commaless. So this gives the idea कि, but if suppose if there is a comma, if we consider the comma in between, the results are not correct. The result found in the protein, they are not correct. Suppose one of the base is wasted for saying stop, first amino acid has specified, now U is wasted, the second base is wasted, for example C is wasted for this thing, and the second combination will be CU. So next one, that is CGU, like that. The resultant protein formed do not show the amino acids in this in the correct sequence, and that's why possibility of comma is not there, and call the characteristic feature of genetic code as a commaless, or code is commaless. Then we will go towards the another property, that is ambiguity of the code. Genetic code is ambiguous, or ambiguity of the code. Ambiguity means doubt. So there is no ambiguity in the genetic code. The code is certain in the sense that specific triplet codon codes for specific amino acid. Each triplet codon codes or specifies always or at all time the same amino acid. For example, codon UUU that we have seen. So it codes for phenylalanine, and it codes for phenylalanine always, and it will not code for any other amino acid under natural conditions. However, UUU may also code for other amino acids. For example, in presence of antibiotic streptomycin, UUU may also code for amino acid isoleucine or leucine or serine. So here, thus when codon codes for more than one amino acid, means UUU codes for phenylalanine, and at the same time when it comes in presence of antibiotic streptomycin, it also codes for the amino acids like isoleucine, leucine or serine. And when such thing happens, means when codon codes for more than one amino acid, such property of genetic code is called as a ambiguity. So higher magnesium ion concentration changes the pH, and addition of substances like ethyl alcohol in the cell also result in the ambiguity of genetic code. Means if such situations are there, like addition of ethyl alcohol or in the cell surrounding, that can cause the ambiguity of the genetic code. Okay, next property, that is the code is degenerate. Genetic code is degenerate. Code is commaless. Code is ambiguity of the code, that also we have seen. Universality of the genetic code, that also we have seen. Now the another property, that is code is degenerate. See, remember these properties, they are asked for full answer question for many times. The code is degenerate. So we know there are 64 triplet codons that we have seen just now in the genetic code dictionary given by Dr. Hargovind Khurana. So out of these 64 different combinations, 61 codons have been shown to code 20 different amino acids in the cell, and they take part in protein synthesis. Out of 64, 61 they specify 20 different amino acids and are responsible or take part in the process of protein synthesis. Then it is obvious that number of triplet codon is more than that number of amino acid types. Amino acid types are only 20, and number of codons, they are 64 different. Out of those, 61 are responsible or take part in specifying 20 different amino acids. Means it does not mean that most of the amino acids involved in protein synthesis, they have more than one triplet codon. Means it is no doubt there is this possibility more than one codons are there for specifying particular amino acid. When one amino acid is coded by more than one triplet codon, such property of genetic code, you call it as a degeneracy of genetic code. One amino acid is coded by more than one triplet codon. This property of genetic code is called as a degeneracy of the genetic code. Take one example of such type. So see here genetic code dictionary again, and we will go towards taking the example. For example, amino acid phenylalanine is coded by two degenerate codons. Phenylalanine. See here, we have seen it. See these two different combinations. What is the first? UUU. And what is the second? UUC. So like this, these two codons, why they are degenerate? Because they specify only one amino acid, that is phenylalanine. For specification of phenylalanine, the two different codons are there, and as they are more than one, these codons, they are called as the degenerate codons. So like this, in the genetic code dictionary, you can see here, these are the four different combinations for leucine. So I will not take the names which are the combinations, but just see all these four combinations, they specify leucine. And you try to see if this leucine is there in the somewhere in the genetic code dictionary, somewhere else. I don't think that leucine is having. See here leucine. So these two different codons are also there. Means in all, for leucine specification, these 2 plus these 4 plus 2 means 6 codons are there for specifying only one amino acid, that is leucine. So like this, this property is called as a degeneracy of the genetic code. One more property we have seen of genetic code. And one more property we will have to see here, that is code is non-overlapping. Code is non-overlapping. What did it mean? So don't suppose this is the thing. This is the mRNA sequence on the this is the sequence of basis on the mRNA. Okay. Just see. See here. If we take AUG CGU, for example, 1, 2, 3, 4, 5, 6, 7, 8, somewhat like that. Okay. And take the CUG. Only 6 bases at a time. Okay. So if we read in a continuous fashion or in a non-overlapping manner, how many codons will be there? AUG1 and CUG second. If you go towards the genetic code dictionary and search for AUG codes for alanine, AUG codes for alanine, or sorry, AUG will code for one amino acid and CUG will code for another amino acid. Is it clear? Okay, this is the situation if the code is read in non-overlapping manner, and if it is read in a overlapping manner, so here we can say AUG, then take start from G again, GUC, next 3 like this, then take start from this GCU. So like this, how many combinations? More than 2 amino acids, 3 codons here, and more than here 3 codons, they will specify 3 amino acids. But this is not the situation exactly or actually. When under experimental condition, if one say the formed polypeptide chain, it contains only 2 amino acids in it. It contains only 2 amino acids in it. And that's why we say that the genetic code is non-overlapping. So like this, we have studied Like this, we have tried to study the chapter named as a genetic code. So most probably, for most of the time, the characteristic features or properties of genetic code are asked in the examination.